Hello, everyone. Thank you all for coming. My name is Eric Lassiter, and yes, I have been blessed to spend the last 8 weeks in Dr. Ferreira's lab, where I've been able to learn about pituitary adenomas and them specifically at the single cell level. So a little bit of background on pituitary adenomas is that they are these benign tumors arising from any of the 6 major cell types of the pituitary. And despite being benign, they can present with these severe comorbidities due to this hormone oversecretion from the tumor. And one of the biggest things that I want to highlight is that within the literature, they are thought to be monoclonal in origin. However, this can raise some questions when we start to look at the treatment of these tumors, and specifically at the current chemotherapy agents, which are currently only able to sort of suppress this hormone oversecretion, but are not really able to promote any cure or prevent recurrence. And it's because of this lack of efficacy that they tend to be sort of the last resort of treatment, secondary to surgery and radiotherapy. However, once surgery and radiotherapy fail, patients are often found in this sort of battle between the symptoms of the tumor hormone oversecretion or potential side effects and the cost of prolonged chemotherapy usage. So within Dr. Ferreira's lab, in this pursuit to identify better molecular signatures of these tumors in order to promote more targeted novel therapies, they came across this hypothesis that these tumors aren't actually monoclonal as they are conventionally prescribed. And if they were able to find a heterogeneity of tumor gene expression, it may explain the lack of efficacy for the current chemotherapeutics that are available and help us to guide help guide us to have better treatment approaches in the future. So in order to identify heterogeneity within a single tumor, the lab employed this methodology. It's called single nuclei RNA sequencing, which works to profile the gene expression of single cells by isolating the nuclei from these cells and then sequencing the mRNA that is found within. It is extremely advantageous, obviously, because you are able to compare the cell-to-cell transcriptomics within a single tissue. And if you find these differences, then you know that it would not be monoclonal. So the portion of this procedure that's done within the lab entails sort of the sample preparation. Isolated nuclei from frozen tumor tissues are partitioned into these little oil droplets that will also contain a single bead, a single gel bead. And these gel beads contain the necessary probes in order to bind to the mRNA, complete reverse transcription, and then also add these barcodes and adapters and indices for sequencing and then also downstream nuclei tracking. So once the complementary DNA has been synthesized and then amplified to an adequate concentration, it is then sent off to Northwest Genomics for the sequencing portion. And so once the sample libraries are introduced into the flow cell in that top left photo, the forward and reverse strands of the complementary DNA template are synthesized and then amplified via bridge amplification to generate these clusters. And then the actual sequencing portion occurs in these cycles where these fluorescent these nucleotides with fluorescent dye tags are introduced. And as they bind in complement to the strand, they emit this light, which has a very characteristic wavelength and intensity that can be recorded. And then after all of the reads have been sequenced and the recordings are converted into their respective nucleotide bases, we here at the lab will receive all of the reads and then process them through various statistical analysis pipelines, which work to properly align the reads, filter out the poor quality data, be able to scale it, perform this linear and nonlinear dimensional reductions, complete biomarker testing on the clusters, and then also do some cell type annotation. So these figures here that I'm showing you are from an actual pituitary tumor tissue that was sequenced. In the top left, we are looking at the before and after of the data within the quality control step of the pipeline. In the top right, we are looking at the linear and then nonlinear dimensional reduction step of the pipeline, which you can already see and appreciate sort of the different clusters within this tissue that are showing a difference in gene expression. And then on the bottom portions here, in the center is the cell type annotation portion, which is identifying the specific molecular signatures of these different cells within the one tissue that are different. And on the bottom right here, we're looking at a series of genes that are upregulated in some clusters while not in others. And so what all this shows us is that it provides evidence against, or it provides evidence to show that not all pituitary tumors are monoclonal in origin, and also provides a series of potential gene targets for therapies in the future. And then I want to thank so many of you for such an unparalleled opportunity and experience that you have blessed me with this summer. Dr. and Ms. Elbogen, thank you so much for having the vision and compassion to start this program many years ago. Thank you to Dr. McDonald, Jim Pridgeon, the entire selection committee for seeing the passion of neuroscience within me. Thank you to Dr. Ferreira, Dr. Parada, Dr. Duvari, and the rest of Dr. Ferreira's lab for being such generous hosts and mentors to me. Thank you to Sylvia and Julie for all of the hard work that you have placed into creating such a meaningful experience for us students. And thank you to the entire neurological surgery department, the donors of this program, and the rest of my 2022 cohort. Thank you.